Good morning and welcome. Thank you for being here, and special thanks to the members of the committee for accepting being here as well. My name is Carlos Toscano Ochoa, and today I'm going to present my thesis called Principles of Distributed Computation in Non-Growing Cells, developed at the Dynamical Systems Biology group, led and supervised by Dr. Jordi Garcia Ojalvo. The main outline of my thesis, I will start discussing the introduction of the project. Then I will point out the purpose. I will introduce the concept of what a wire is, what it should be, its architecture, and then I will move on to wires in the stationary phase. We will discuss later the mathematical model. We will explore together the model and analyze its temporal dynamics. We will discuss how it behaves when we have digital periodic inputs, and finally the conclusions. So let's start. This shows one of the main point of views of or objectives of synthetic biology, which is trying to develop synthetic tissues or engineered cells, tissues or cells, by combining proteins and genes in an incremental complexity until we reach that stage. Now, this figure, it's the way of thinking in synthetic biology, in the sense that we expect synthetic biology to behave like engineering, in which we have some physical components that we can connect them so they behave as, for instance, logic gate. We can integrate that into circuits, and then we can build computers. There was an idea of, instead of being inspired by engineering, maybe we can emulate engineering. So why couldn't we be able to implement something like a chip? After all, a chip in its inside is just a bunch of logic gates connected among them. And actually, we can develop biological logic gates. In this example, for instance, you have how to create an AND logic gate. These circuits implemented in bacterial cells, if you have IPTG and arabinose simultaneously in the culture, these two substances will induce the expression of these two transcription factors, R and S, that when they are both present at the same time, they will induce the expression of the green fluorescent protein, GFP. Then the host cell will become fluorescent. And actually, we can see that it works, even only if both arabinose and IPTG are present at a high concentration simultaneously, we can see that there is a peak in fluorescence. So we can, if we can implement single logic gates in the individual cells, couldn't we put all these logic gates inside one single cell and then force that cell to act in the same fashion that this chip does, for instance, which is, by the way, an arithmetic logic unit. It's a component present in every computer. And well, not so fast, because maybe genetic circuits, gene circuits can alter or affect gene expression. In this experiments by Cerroni et al., published in 2015, they had E. coli, and they inserted a GFP gene, synthetic gene, in the genome, just only one copy. So the cell will be filled with GFP protein, and it would become fluorescent. And if we see the measurements of fluorescence 1 hour after how that fluorescence has increased, we got these measurements. Now, if we add a plasmid, an empty plasmid inside the cells, we can see that the fluorescence that we observed now is a little bit, has decreased a little bit. If we put inside a plasmid with an inducible set of genes, an operon, it decreases even more. And if we induce, by adding arabinose, this gene, this operon, and now we have all these proteins, it keeps decreasing. So we can see that genetic circuits, gene circuits, can actually impair and negatively affect gene expression. That's a problem. What about growth? Well, here you can see from the same paper two growth curves measured in optical density at 600 nanometers of wild type E. coli plus this E. coli that it only has one single gene, right? The DFP gene, and they behave more or less the same. It seems that it's not a big issue, but as soon as we compare it with the growth of E. coli hosting a plasmid with induced or not, we see that there is a decrease in the growth rate. So yes, gene circuits can negatively affect growth. Now, does it mean that we cannot anymore create complex circuits inside one cell? Well, certainly we have the problem of negatively affecting gene expression, and also the problem of negatively affecting growth. So if we have a culture composed by a single cell type with the whole circuit inside, and this may present these problems of the growth and gene expression, it appeared an idea of splitting the circuit and hosting the different, for instance, logic gates in different cell types, different strains, and connect them using chemical wires. So at the end, we would have a culture composed by a homogeneous mix of these strains, right? And this was an idea that was proposed 10 years ago, which is called distributed computation. Now, chemical wires, what are these? A chemical wire is it's an entity that we will discuss that is based on diffusible signals, chemical signals in the media, that actually makes one of these strains behave as an emitter of the signal, and another strain act as the receptor of the message of the signal. And this has been commonly implemented using quorum sensing system from gram-negative bacteria. In this case, what you do is you split the system and place for instance, the LuxI gene responsible for the biosynthesis of acyl homosyryl lactones, tiny diffusible molecules that can be sensed by a receptor, another receptor strain, in which you place the LuxR, which is a transcription factor that in the presence of acyl homosyryl lactones, if the concentration of these tiny molecules it's above some arbitrary threshold, then it can induce gene expression, right? Now, there has been several approaches to this distributed computation approach, right? But the thing is that they have been mainly focused on how can we create a network, for instance, using distributed approaches, or logic gates, but no one has even focused in something more elementary, which is, okay, what about the wires? What an ideal wire should be like, right? Or we are mixing several strains in a culture. Should we expect the same behavior if we alter the strain proportions? What about the temporal dynamics of a wire, how it behaves under different circumstances? Can we expect any interesting emergent behavior of something so boring like a wire? And even more, if that is the case, could we use a single wire for some specific task? So moving forward with the wire architecture, this is the main architecture that you can observe mainly in most of the iGen projects. You only have one cell type, one strain that is going to be the emitter, that in presence of input signal is going to secrete these communication molecules. Now I'm going to keep it abstract and general, so that I'm going to call this any communication molecule biobits, shortened BBIT. And the thing is that these molecules will be sensed by another strain, which I will call the receptor strain. I see a problem with this architecture, and that is that if I remove the input, I can depend, it might depend on the half-life of these molecules for the receptor to turn off its response. So I propose adding a third strain, which I call the zinc. So that third strain will be responsible for the degradation of these biobit molecules in the media. Now, what about the architecture of these three strains? Well, the emitter must have the minimum required set of genes for expressing biobits, for synthesizing and releasing to the media biobits when exposed to an input. A receptor must have the minimal set of genes constitutively expressed required for sensing this biobit concentration and induce output transcription. And the zinc, again, must have the minimum set of genes required for biobit degradation, right? And these would be also constitutively expressed. So what about the biobit, okay? What is a biobit? Well, a biobit must have, in my opinion, 6 desired properties. It must be biosynthesizable. It must be also secretable. It must have a long half-life, but at the same time, you must be able to degrade that biobit enzymatically. It must be biodetectable, and also it must be non-toxic for the host cells. We could, for instance, put an example, which is the 3-oxo-cis homosyl lactone. It has been widely used, and it can be synthesized by LuxI. It can be sensed by the transcription factor LuxR. It can be degraded by AIIA enzyme from Bacillus species, and at the same time, it has a long half-life. In this case, days. Very convenient if you expect your circuits or whatever you want to act in hours. It diffuses to the membrane, so it self-secretes itself to the media, and it's not only non-toxic, it's not toxic, but if we use this specific E. coli knockout and we remove the gene ASD IA, this specific strain is totally blind to lactones, so it cannot upregulate or downregulate gene expression. Now, for wire implementation, specific wire implementations where I choose LAC-I as the input sensing system. So the emitter strain will interpret IPTG as input and will produce biobit 3-oxo-cis HSL with a gene LuxI. The receptor, this will be the structure. So the receptor core is the required machinery to sense that biobit, and the output would be GFP. So we will interpret GFP fluorescence as the output. And the zinc strain will have AIIA enzyme constitutively expressed, and it will be responsible for biobit degradation, for lactone degradation. And the 3 constructs will be placed on PSB1C3 plasmid from the IgM competition. Now, we know that if we want to have a complex circuit inside one cell, we're going to have problems with gene expression, but this could be overcome with distributed computation. But what about growth? Well, if growth can be impaired or affected, negatively affected, well, I propose you to forget about cells that are growing and work in non-growing cells. So here you can see our receptor strain fully induced in LV media, and you can notice that GFP fluorescence increases even when cells have arrested the growth. So okay, that is a good point. Okay, there's something positive. They express GFP in a stationary phase. Now don't get overwhelmed with this and follow me first. What happens with the host cell without any plasmid if we expose to a different concentration of biobit in LV media or in Davis media over time? And we are going to measure optical density at 600 nanometers, so cell concentration. We can see that that E. coli grows slower in Davis media, which is a minimal media, in this case supplemented with 0.4% glucose. But BioBit concentration does not affect growth of the host cell. Now, if we see what happens with the receptor strain, now you can appreciate that even though we get some fluorescence in both cases, in LB and Davis media, it turns out that it negatively affects growth. And we know it cannot be due to BioBit, high concentration of BioBit. So GFP expression inhibits growth. Okay, that's something that we don't want to have. Let's switch to another strategy. Instead of inducing our cells directly from the inoculation, let's wait, let's inoculate, let's leave the cell, the culture reach stationary phase, and add BioBit 24 hours or 48 hours after inoculation. So these two specific time points will be considered the time equal 0 hours in these 4 plots. And as you can see, Davis media seems to make cells behave properly. They keep their optical density pretty constant across 50 hours. And if we take a look at what's going on with the fluorescence, well, in the case of LB media, both in 24 and 48 hours, we see that there's a peak in fluorescence. And we can also observe peaks in the case of Davis media. So that's something that we don't want. Nevertheless, if we add the BioBit, in this case, the lactone, 48 hours after induction, we see that there is a nice induction. And so if we use this E. coli strain from the KO collection, that is lactone blind, it does not respond to lactone, and we use Davis media with 0.4% glucose, and we induce ourselves 48 hours after inoculation, which means that they are in a stationary phase, what we get is a stable optical density, a constant rate of protein synthesis for hours, in accordance to this work by Geffen et al. From 2014. And the fluorescence seems to reach equilibrium in less than 40 hours. So, okay, we have conditions for testing these wires, the stationary phase. Now, let's talk about the model. It's extremely simple in this case. I'm going to assume that the rate of BioBit synthesis is going to be proportional to the amount of emitter, of the concentration of emitters in the media. And also I'm going to assume that the BioBit degradation is going to follow a Michaelis-Menten dynamics. So meaning that for low BioBit concentration, we expect that the degradation is going to be more or less proportional to the concentration of BioBit. But if BioBit is present at high concentration, then the degradation is going to saturate. And that process is going to be proportional to the amount of zinc strain that we have in our media. And recall that because emitters are inducible, you can induce the emission of BioBit, we are going to have two different emission constants in the case of emitter strain. One of these emission constants in the absence of input will be close to 0. Now, what are the model assumptions? We are going to assume that the abiotic degradation is going to be negligible. We are going to assume that also we are not going to have any problem with the BioBit precursors. The emitters are going to instantaneously switch from uninduced to induced when exposed to BioBit. Also, the receptors are not going to participate in the synthesis or degradation of BioBit. And also, they are not going to hijack BioBit molecules. It's going to have a really very dynamic binding and binding with BioBit molecules. And the output production from receptors will be an instantaneous function of the BioBit concentration. Now, this is an ideal wire. This is like the minimal, the most ideal scenario. So this model is intended to be something that if the model tells you that something does not work, that some implementation is not going to work according to the model, then it doesn't matter if you make more refinements to the model, it won't work. And if the model tells you that it might work, so it's worth trying adding more complexity to the model. So if we have a culture composed by a population with concentration P measured in cells per milliliter or in corrected optical density at 600 nanometers, we can decompose that in an amount of emitter cells, and an amount of zinc cell, and an amount of receptor cells. But actually, we are going to be working with the ratios, the strain ratios. Epsilon for the ratio of emitters in the whole population, sigma for the ratio of zinc in the whole population, and rho for the ratio of receptor in the whole population. And the ratios will remain constant, and they add up to 1 for whatever combination of the 3 strains. Now we have a new problem. We have an infinite possibility of different combination for zinc, emitter, and receptor strains, to combine them, to mix them together. How does it affect the solution of our model? And I'm going to take advantage of a particular theorem, geometric theorem, which states that if you pick a point inside a triangle and you add the distances to the 3 sides of the triangle, that sum is going to remain constant, no matter in what part of the triangle, of the inside of the triangle, you have chosen your point to be. That is cool, because that means that we can express every possible combination as a point inside an equilateral triangle. And well, cool. We have a bijection between all possible combinations of zinc, emitter, and receptor cells with one single point inside an equilateral triangle. And now we only have we can map the points inside an equilateral triangle to the solution of the model. Now, recall that we are going to just affect the ratios of our strain, the proportion of emitter and zinc strains, and also receptor, but the other parameters are going to remain fixed. So if we homogeneously distribute some points, each of them representing a unique combination of emitter, receptor, and zinc strain, and we run some simulations, okay, we can appreciate that some of the biobit time traces, biobit concentration time traces, they reach equilibrium, whereas other they tend to diverge. So if we review where are these cultures, we see that all the cultures that are associated with convergence biobit concentration and reach equilibrium, they are placed in a specific place of the triangle, whereas others are in another specific place of the triangle. So let's focus on the convergent trajectories. The first thing that we should do with convergent trajectories is finding the fixed points of our model. For that, we assume that there is no change, that the equilibrium has been reached, and then we can solve for the biobit concentration at equilibrium with that expression. And because a concentration must be positive, this inequality must be satisfied. The ratio of emitter over zinc strain must be smaller than this ratio that comprises 2 different parameters of the model. This has to do with the fact that the denominator of that fraction must be a positive number. And that is the condition for convergence, which allows us to make sense of what's going on in the different regions. For instance, in the region in the left, which is called the convergence region, the conditions for convergence is satisfied. Whereas in the other region, the divergence region, it's not satisfied. The ratio of emitter over zinc is written at this specific ratio. And the line, the segment that separates both regions is called the horizon of divergence. Now, one may ask, what is, if we are placed in the convergence region, can we find a geometric place such if I have 2 cultures or 2 points placed in that geometric place, it is ensured that they will reach exactly the same biobit concentration and equilibrium? And the answer is yes. Any segment irradiating from the top vertex to the bottom side of the triangle, it's ensured that if you have cultures, that they will reach exactly the same biobit concentration at equilibrium. something that you can, for instance, notice with the circles. They all reach exactly the same. They converge to the same bit concentration at equilibrium. The same happens for the squares, and the same happens for the diamonds. And that is given by this expression, as I told you before. Now, in the case of divergent trajectories, they tend to asymptotically approach a linear divergence. So the isolines of asymptotic divergence rate, there happens to be lines that are parallel to the horizon of divergence, and you can see that all the culture circles, for instance, that lie exactly in the same isoline, they tend to diverge with the same rate. The same happens for the squares, and the same happens for the diamonds. And the slope of this asymptotic growth, asymptotic linear growth, is given by this expression, that is the asymptotic divergence rate. Okay, what about the receptor strain? The receptor strain, we're going to assume that it has just a generic transfer function, sigmoidal, but in this case, just imagine a generic Hill function. And we're going to normalize, work with the normalized response of the receptor strain. And for instance, if we have a biobit concentration that is above what I call the threshold 95, it's an arbitrary threshold, we will say that the receptor is fully induced, meaning that the response that we can measure is above 95% of the maximum response that the receptor can give to us. Also, if the biobit concentration is in between these two arbitrary concentrations, threshold 5 and threshold 95, we will expect a variable level of induction of our receptor strain, meaning that we will get a response in between 5% and 95% of the maximum possible response. And finally, if our biobit is from 0 biobit concentration up to the threshold 5 concentration, then we will say that this is associated with that our receptor is totally uninduced, right? Because the signal is less than 5% of the maximum response. So, because in the convergence region, now here plotted in color in the triangle, each point is associated uniquely with biobit concentration at equilibrium, we can map that biobit concentration to the normalized response of the receptor, and we can see where this uninduced receptor, variable level of induction, and fully induced receptor, where this region, where are these regions in the convergence region, right? Also, we know that in the divergence region, because the biobit will keep increasing, at some point, our receptor will be fully induced. So here we care more about the asymptotic divergence rate. And because a culture entirely composed by emitter, strain, and fully induced will have a maximum divergence rate, we can normalize any divergence rate by that amount and get, again, a value between 0 and 1. Okay, so the wider space can be represented using this triangle representation, and gives us meaningful information about how it behaves. We have measured the induction of our receptor strain, and what we did was induce our receptor strain 48 hours after inoculation in Davis media, and you can see that it reaches equilibrium, and we did some fittings of a generic Hill equation with leakiness. You can see that leakiness is almost 0 in this case, and the maximum response is going to be normalized. So at the end, we only care about the Hill coefficient and K value because we're going to be working with it normalized. And these experiments were very reproducible, and we could have had good measurements of our receptor strain, of our parameters. We also perform fittings of 3D solutions to capture also the time dynamics of the receptor using another approach. At the end, you see that it converges to this sigmoidal shape. Okay. Okay. And I've been showing you what happens when there's input in the media, and you get a lot of variability in the media, which has been associated with this representation. But as I told you at the very beginning, you may remove the input, so the emission constant will be close to zero, and you will have almost zero variability in the media produced by the emitter cells. And it turns out that this is associated with another representation of the wire space. In this case, this scenario means that no matter where you choose to your culture to have this proportion of, no matter the proportion that you choose for your wire to have in between in zinc receptor and strain, the composition, the proportion of this, your receptors they are not going to be induced, which is reasonable given this scenario. These two are the wire states, and in particular, these are the proper states. Okay, so now if we recall that even when we have these two states of the wire state of the wire space, if we have a culture that is lying in that precise position, it's in the same position in both scenarios. We are only changing all the horizons inside the wire space. So now I'm going to define some new regions. For instance, this first region, it's the intersection between the induced receptor region when the emitter is induced and the uninduced receptor region when the emitter is uninduced. And this is what I call the digital region. Okay. Now, there is another relevant region, which is the intersection between divergence region when the emitter is induced and uninduced receptor region when the emitter is uninduced, which I call the buffer region. And these two regions, it's what I call the operative regions. And these regions have some desired properties if we want to do distributed computation. So you want your strain ratios to fall in these regions. So let's see how these regions behave. If I have a wire placed in the digital region in green, and we apply some inputs in maybe short input, maybe long inputs, we see that the response of the receptor is more or less the same as the input signal. So actually, the output is more or less the same as the input. Whereas if I place a culture in the buffer region in pink, it changes a little bit. We see that the time that it takes for the receptor strain to turn off has increased. And okay, we can say that the output duration is greater than the input duration if our cultures are there. Now, let's dig in the temporal dynamics. Let's start with definitions. The input time here, it's just the duration of the input, whereas the output time, I'm going to consider that it is all the time span in which the response of the receptor strain is considered to be totally induced. Okay, and these two quantities, the T sub on is going to be the time that it takes for a wire to turn on the receptor strain, and the T sub off is the turning off time. It's the time that it takes for a wire to turn off the receptor strain, starting from the moment that the input disappears from the media. So first, what about the turning on time? Okay, which would be this time span here. Well, if we analyze this turning on time, we see that, for instance, this is what we get, and we see that wires that are close to the top vertex, they will increase its turning on time, meaning that we will require our input signals to last at least that amount of time, so we can expect some response, any response from the receptor strain. Whereas if we are close to the bottom of the triangle, we are minimizing that time. So our wire could be work in shorter time scales. Now, what about, oh, sorry, I'm going to show you here a video of what I'm telling you right now. This is a sample of cultures that are moving across one of these ISO lines that irradiates from the top vertex to the bottom. So they all are meant to reach exactly the same biobit at equilibrium concentration. In this case, it's 5 arbitrary units of biobit. And you can see that for most of the cases, for most of the culture, the response will reconstruct it. But in the top part of the triangle representation, you would have needed a longer input signal for the response to even be considered as induced. Now, what about the turning off time? That time span right there. Well, I told you that this is the time that it takes for turning off a wire when starting with counting from the moment that the input signal disappears from the culture. And it turns out that this turning off time, is proportional, okay, to the input duration, okay? And this scaling factor depends actually on where your wire is placed inside the wire space. So it depends on the specific proportion of the three strains. And actually, what ideally the minimal value that we could expect for this scaling factor is 1 if our wire is placed in the digital region. So that would be perfect reconstruction of the input signal in the output signal, or this scaling factor if our wire is placed in the buffer region. That you can see that depends particularly in the ratio of emitter over sink strain. And this is the better that you can get. Okay, that is the ideal situation. So you can see what happens with the turning off time if we place some cultures, for instance, in 3 cultures in the digital region and other 3 cultures in the buffer region in pink, and see the turning off time. In the case of the cultures placed in the digital region, you can see here, okay, that it's not necessarily 0, and the closer that you are to the top vertex of the triangle, you can see that it starts increasing the turning off time. But if for the cultures, but these quantities remain fixed, and it does depend on the input time, the duration of the input signal. But if the cultures are placed in the buffer region, that's another story, and you can see that how the turning off time starts increasing in a linear manner. Okay, starts increasing in a linear manner, and it depends actually on the input time, right? So whereas this time for the wires in the digital region, I call it lag time, and it does not depend on the duration of the input. The other case, cultures that are lying in the buffer region, I call this a buffer time, right? And this is very important because that tells us that buffer region can store information about the duration of the input signal. So buffer region can, in fact, act as a timer. Okay, let's see the difference in cultures when you have a culture that has exactly the same amount of receptors strain. Okay, in this case, the distance from this point to the bottom side of the triangle would be the amount of receptor cells. So this is constant in any case, and we are only changing the amount of emitters and sink strains. And you can see that when we are in the buffer region, the biobit tends to increase and decrease in a linear manner, expanding the response time, the output time. Whereas in the digital region, we get a good reconstruction of the input. Okay, something really important. This triangle in the left is what I've shown you. For instance, this could be the case of wire fully induced. Okay, this is we have biobits in the media, so the wire is fully induced. If we put a culture there in this point, this specific point, we know that our receptor strain is going to be fully induced. But at the same time, the proportion of receptor cells that we are going to expect in this part of the triangle is going to be very low. Actually, it's going to be less than 10% of the culture. So even if these cells are fully induced and they have high fluorescence, maybe you are not able to sense that just because they are underrepresented in the population. Okay, let's go for the solution. Let's move our culture to the top vertex, closer to the top vertex of our triangle, and here you expect at the equilibrium exactly the same level of induction of our receptor cells, right? And they are more than 80% of the population of the wire, okay? Because they are close to the row vertex, to the vertex that represents the amount of receptor cells. So we should be able to get good measurements. But wait a second, not so fast, because the fact that you have placed your culture close to the top vertex means that it will take longer for the receptor strain to reach equilibrium. Okay, so you would need this would be an ideal case if you're expecting the time scale of your inputs to be long, right? So that tells us that this time response interplay is pretty interesting, because if you're close to the top vertex of our triangle, the response is going to be slower but easier to detect, right? But if we move, if we want our wire to be more ideal, then we need to move to the bottom of the triangle. That in that case, our wire will be faster, but also we will have to face the difficulty of detecting, sensing the responses. Now, what about digital periodic input signals? I'm going to call this in periodic digital input signal when the presence of an input repeats itself with some period. And actually, the duty cycle it's something really important here. It's the fraction of a period that corresponds to presence of input. Now, for instance, if we take the daylight hours per month, okay, in Barcelona, we can then obtain, by dividing by 24, the duty cycle associated with each month. Okay, so that would be the duty cycle of each month of daylight hours in Barcelona, of every month. Sorry. Now, pay attention to this plot. It has a lot of information. In the top left part of our plot, you can see that we have a periodic input signal, okay, with time increasing in the y-axis. This part specifically will be associated with an induced wire, an induced emitter, and this other part will be associated with fully induced emitter, okay? So imagine that now, focus please in the triangle in the bottom side. Imagine that we choose a culture that is there, meaning that there is no emitter cells in the media because we are super far from the super far from the vertex associated with the amount of emitter cells. This would be a wire only composed by zinc strain and receptor strain. If the culture is there, this strain, this straight line, vertical line arrow in red shows the time series of response, the response time series. So in this colorful plot, I'm just showing you what happens with all the possible cultures lying in this segment A to B, right? And the color encodes the normalized response. And you can see that for all the cultures that are lying in the digital region, we get a good reconstruction of the input signal in the output, in the response, right? Whereas in all the cultures that are lying in the buffer region, we see how the duty cycle tends to increase. And on top of that, if we have a culture placed here specifically, it turns out that we lose the periodicity of the input signal in the output signal. So we have been able to filter in the cultures that lie here in this specific part of the buffer region all these signals, or these periodic signals, increase its duty cycle until the signal gets totally filtered out. So that means that we can filter periodic, we can modify or alter the duty cycle of the input signal in the output, but also we can filter these signals. And we can analyze actually where are the filter horizons. For instance, these are 3 examples of input signals. The first one with a duty cycle of 90%, the second one with a duty cycle of 50%, and the last one with a duty cycle of 10%. Now, if you want to filter signals with a duty cycle of 10%, for instance, you need to move closer to this this side of the triangle, okay, the side more closer to the right. And if you have to filter signals with high duty cycles, you don't need to go so close. The filter horizon is going to be more it's going to be farther from that specific side of the triangle. Now, if for instance I feed an input signal with a duty cycle of 20%, and in this case I have a wire with this specific emitter to with respect to sink ratio, okay, which places our culture in the buffer region, okay, we see that we are in this region where periodic signals are never filtered, but we are in the buffer region, meaning that the duty cycle of the output is going to increase with respect to the duty cycle of the input, which is exactly the case here. Here, the duty cycle has increased. Now, if we instead supply some other input signal, in this case with a duty cycle of 65%, and we see what happens with the red, the culture in the culture with the red dots, meaning that it is exactly the same ratio as emitter and sink. Okay, it's exactly the same ratio of emitter over sink, but in this case, because we have increased the input, the duty cycle of the input, okay, we are now in a region where periodic signals are always filtered. And that means that we lose the periodicity of our input signal in our output signal. Finally, we could change the ratio of emitters and sink, and now this is the blue one is the new scenario. In this case, this is placed in the digital region, okay? And in the digital region, what happens is that you're not going to amplify, ideally, your duty cycle. So you can see that the blue output signal has more or less the same that the input signal, the more or less the same duty cycle, excuse me. So the Y parameters allow us to define regions when we can always filter our input signals. Now, this is a video showing you what happens with periodic digital inputs. For instance, if we move into the digital region, okay, we restore the input in the and the output in the we restore the input duty cycle in the output signal. That you can see it right now. If we move closer and we go inside the buffer region, what happens is that biobit concentration tends to increase. You're going to see it right now, to increase and decrease in a linear manner, which increases the duty cycle, and even it can eliminate the periodicity of the response of the output signal. Now it's time for a final divertimento, an illustrative example of what could we do with something that sounds so simple as a wire. Could, for instance, a wire respond to subtle changes in the amount of daylight hours? Well, this is the table that I showed you before, the amount of daylight hours in Barcelona per month with the corresponding duty cycle. We can plot these duty cycles like this, okay, showing the duty cycle for each month in Barcelona. And if we place our culture wire exactly there at that point, a point that we can calculate its coordinates, we ensure to filter precisely these 3 months. Okay, so that would be the input. Okay, that would be the input signal. It has some periodicity. In this case, it's each month it's going to have just only 5 days. We don't need more. And maybe you can notice that the months of the middle of the year, they're a little bit thicker. So if we see what happens with the biobit concentration, you can appreciate that precisely months 5, 6, and 7 that are above our threshold, okay, these are the ones that make the biobit concentration increase on average. And when we reach August, which is below the duty cycle threshold, then the biobit concentration tends to decrease on average. Now we see what happens with the response. We can see that we have been able to obtain, we have been able to filter the periodicity of the daylight hours in the receptor strain, which is cool because we don't need a circuit, we don't need to sense, for instance, temperature. Which this just a wire, just a single wire is able to sense subtle difference in daylight hours. So yes, we can make a wire to respond to these subtle differences. Now, with the permission of the committee, I would like to read the conclusions of my thesis. First of all, we propose a chemical wire architecture for distributed biological computation. Also, we present an experimental setup for testing genetic circuits in a stationary phase in E. coli. Third, our model predicts how input signals can be restored or modulated in the output. Four, chemical wires can store temporal information and the system can act as a timer. And finally, digital periodic input signals can be filtered by altering the strain ratios. The theoretical results of this work has been published this month in iScience with the name A Tunable Population Timer in Multicellular Consortia. And now, thank you very much first to my supervisor, Professor Dr. Jordi García-Orjalvo, and to all my colleagues of dynamical systems biology, the current and the former ones. Thank you so much for being here, and I will be more than happy to answer all of your questions. Thank you very much.
All right, thank you. Thanks, Carlos. You can stop sharing screen.
Oh, yes.
So we can see each other. All right, very good. Okay, so we'll proceed to the questions from the committee. We shall start with Ángel Goñi Moreno from the Universidad Politécnica de Madrid. Ángel.
Sure, thanks. Well, thank you very much, Carlos, for the presentation. And
thank you.
Congrats on the work done. I think it's very clear about everything, and that I appreciate very much. So if the problem is clear, the solution is clear, the background and the approach, everything is clear. No? The text also is very, the narrative is friendly, let's say, so you can read very nice. Okay, so it's not hard to read. So I have a few questions. Okay, I will go through them. Well, first of all, you did both the mathematical and the wet lab work, right?
Yes.
So that's a lot to do, right? Because both fronts are quite complicated in themselves. So how did you manage to do them both? What is your background in each of them?
What is my background, or what I?
Yeah, first of all, what is your background? Because I guess that you will have more difficulties in one of them, no, than in the other.
Well, I'm a biologist, but I cheated a little bit because I started mathematics, and they changed to biology. But I'm passionate about maths, so I never properly took a course to understand how to do this. Just, I don't know. I learned little by little, baby steps. And well, I wanted to develop this both experimentally and theoretically. And well, my skills for wet lab, I was able to develop them during the master thesis and also the PhD. Yes.
Yeah, no, nice. I say because most of the theses I've seen that are interdisciplinary, no? They always have some one of the fields, no? But you actually did a good job on there. Okay, so well, on chapter 1, I have no major comments. That's a very clear introduction to the problem.
Thank you.
If anything, there is a sentence that says, nobody is interested in standardization. And that broke my heart a little bit because I am. Right?
No, but let me explain myself.
I saw the point on that about the IGM competition and the more specific events and all that stuff. Yeah. I see the point.
No, okay. No, yes. Let me explain to the audience that point, the particular point. The thing is that when I've attended a couple of congresses focused on synthetic biology, and I don't see most of the research groups, they are not interested in the problem of standardization. I see that in for instance, IGM competition. That is something that people want to increase and upgrade the standardization of biobricks and whatever. But in congresses, what I've seen is that they are more focused, for instance, in robustness, for instance, or developing new techniques. I'll give you a couple examples. For instance, Victor de Lorenzo, from the group of Victor de Lorenzo, was published a method to make no leakiness, for instance, right? Inducible systems that have zero leakiness. And the results are astonishing. This, when they say it's zero, it's 100% zero. Even if they were expressing an enzyme that just only one enzyme kills the cells and they were alive. So they are not focused in the standardization. It's fine. It's fine. We need more robust approaches to some of the things that are out there, right? So yeah, in the congresses, I've seen that it's more about improvements, new techniques, new approaches, rather than standardization. But it's not that they are totally separate paths. Yes.
Okay. So in chapter 2, then I have a few comments here. The notion of the wire space, I like it very much. No? The problem of balancing the strains is a problem that we all have in distributed computations. The visualization also, I like that very much. The triangle, it shows very clearly in just one spot the different ratios.
Thank you.
So I just have one concern with this visualization, because in the whole chapter 2, you focus on the emitters and the sink and the dynamics between them two. No? So I found most of the triangles to be just one-dimensional pieces of information. No? So was there actually needed to show two-dimensional triangles, or with one-dimensional light would be enough?
That is yes. One dimension would be enough. You're right, but what do we gain then with the triangle? Where precisely when you try to see the temporal dynamics, for instance, the timing on time, that varies in the vertical direction. So the time that it takes to turn on the wire increases if you get closer to the top vertex. In particular, let me see where the...
Yeah, I know the figure you are talking about there, because yes, it's basically the only figure in which I saw the need for a triangle. Anyway, in the rest of them, I just feel that there is there is more to tell about the receptor. It's not just about the emitters and the sink. For instance, do you think that the receptor would act as a sink itself of some kind? Because by taking the biobits and the receptor will consume biobits itself. No?
Okay.
So I think that I just thought that there would be more to say about the receptor visually. No.
Yeah. Well, the thing is that I wanted to. Well, yeah, you're right. This is the time to turn on the wire. It's the one of the cases, the only cases in which this third dimension, because there is no 3 dimensions, it's 2-dimensional. That way you can see the requirement for this this visualization, and well, playing with the possibility of the receptor straying, for instance, hijacking, or a lot of people have told me over years, why not placing the sink enzyme in the receptor straying? And there's actually a couple of papers that used almost the wire architecture that I proposed. And in one of them, I don't know right now. Okay, in page 121, 122, you have the picture. In one of these approaches, actually is page 119. This is actually almost exactly the same architecture, but in this case, they preferred to make the degradation. The degradation would be induced by the presence of biobit molecules, right? But that introduces some new nonlinearities that I wanted to offer, like the minimal model. It is intentionally a toy model, so you can know what happens when you don't have any relation of feedback or whatever, or this is going to induce its own degradation. No, just I wanted to keep it super clean. So if you implement, for instance, a zinc cell or a zinc cell that it gets induced in the presence of biobit, you can see how different from the ideal scenario it is. Maybe this difference get increased in some regions of a wire space instead of in other in an homogeneous manner. And or what happens if the receptor cells hijack? Can we measure, for instance, how far away from ideality and nonlinearity are we if we start increasing this hijacking? So I think that it's in the same way that with experiments we have a control. I think that we need this kind of model to set a control for other models. I will be more than happy to and I have other results, making this model a little bit more complex. Right?
Yeah, no, I agree with you. It was just a comment on trying to make the most of the triangles, no? Because in that first part, in chapter 2 as a whole, let's say, you just focus on the ratios between the emitters and the sink and leave the
yes,
dynamic implications of the receptors out. No? Okay, so I have in this chapter 2 as well. There is a statement that I would like to discuss with you. You say, what is this? In page 28, you say, but if the minimal wire model predicts that the wire won't work, then it is worthless to test what will happen under more descriptive models. That is something that you said also on the presentation. Do you think that is true in 100% of the cases? So, for instance, wouldn't there be any hidden complexity in the model so that the toy model predicting won't work, but then there are some parameter spaces out there in a more detailed model that turn it into a good solution?
Okay, I think it's tricky to bet for the 100%, especially in biology, but I would say that probably it's worthless to try because, for instance, two of the assumptions of the model is that emitters will switch instantaneously from uninduced to induced when exposed to input. That is an absurd, okay? That is so ideal that is absurd. But this is giving you it's like the best possible scenario. So if you start to add more description in the model and you want, for instance, capture that dynamics, okay, that means that you're going to require more time to reach equilibrium, right? I don't think that under the constraints of this model, I'm meaning that let's build some emitter architecture, receptor architecture, and sink architecture, following this guide that I've written here. No feedbacks, no degradation, degradation that depends on IV concentration. No, just these guidelines. I think that it would be really surprising if I see that something that the model predicts if you measure the constant separately for each strain, and you throw in the simulator and says, hey, you don't have, for instance, a digital region.
Okay.
But it could be, for instance, don't waste more time trying to go farther. But for the same token, maybe you can now know, you maybe know now which are the parameters that you need to increase or decrease. You can make mutations so you can
to focus.
Yes.
Okay. On the parameters, I have another question. In this page 33, you have table 2.1 with a few parameter values. But I think that you don't mention where or why you took that parameter values. It doesn't matter that much, or are they very specific parameter values for this specific?
No, these parameter values they are just for because it is a very illustrative. It's just a sample that gives you nice representation, but it's not so important topologically, in the sense of, for instance, you know that that let me go to, for instance, you are always going to have a convergence and divergence region as long as your emitter constant when there is no input is close to zero, but not zero, right? Yeah. So you know that you're going to have, you're expecting that horizon. So it's not really so relevant, the quantities. I just want to choose some quantities that would show that. Then you know that the bit concentration at the equilibrium that you're expecting in the convergence region is going to increase when you get closer to the horizon of divergence. Is going to increase in a hyperbolic manner, no matter what are your parameters. That is going to be hyperbolic. So, and because it's going from 0 to infinite, then you are expecting your 3 regions. You want your
I think that clarification is
okay.
It's good. No, that you didn't cherry pick the parameters, because then it all works magically.
No, it's going to work with other parameters. So the shapes will change, but everything will keep its relative position.
Okay. Yeah, I liked also very much the regions, the digital and the buffer regions. I think that's a very illustrative way to put all this. Then in chapter 3, I have a couple of questions as well. So, well, first of all, why or didn't you try to simulate some stochasticity in the problem? Did you thought that stochasticity might have an impact on the regions themselves? So,
well, I haven't been doing stochastic simulations because I was thinking about the possibility of meaning. Let me find the proper words, the appropriate words. I haven't done stochastic simulations because stochasticity can be reduced if you have big amount of cells, if the concentration of cells is very high. So I wanted to just solve the problem of cultures in a flask with where there's a lot of a ton of cells that are swimming there. And for instance, if you assume that each individual cell is going to have is not going to have they are not going to share, for instance, the emitters the same emission constant value. Okay, you would expect something like a distribution with its average value like the one that you're measuring. But the fact that all the strains are releasing biobit to the media means that you average all this.
Yeah, okay, so yeah, basically due to numbers, stochasticity wouldn't be an issue in the system.
Yes, right? Yes.
Okay, you mentioned here in chapter 3 several times, you mentioned this protein EL222, which is a blue light using blue light as input, right? You mentioned that several times, and then I expected to see that in the experimental chapter, but I didn't. Did you try it at all? Or?
I started. I have actually I implemented I implemented some of the strains with you know, the idea was to make them produce biobit in response to blue light, and I was testing all the combination of degradation tags, and then we had this coronavirus issue. But I got them there. I didn't have the opportunity to start qualitative experiments, but the idea was in fact to make the cells respond to blue lights, and instead of using for the receptor GFP, which absorbs blue light, use N-cherry, which absorbs like 500 nanometers and emits red fluorescence, sorry. So you can totally separate the emission, the biobit emission, to the measurement of red fluorescence. That was the idea.
Okay, yeah, because it's kind of surprising, no? You read a lot about this protein, and then all of a sudden it disappears. Okay, so fine. Then, yeah, the last part of chapter 3, you describe this non-instantaneous system, right?
Yes.
But it stops there. So you don't run again the simulations with a non-instantaneous system, or it does not make an impact in the definition of the regions or the different dynamic kind of features you have inside?
I just wanted to develop. I didn't do the full analysis of the non-instantaneous wire, trying to define what is the impact of this non-instantaneous behavior in the wire space. But they are required if you want to measure your parameters under some circumstances. For instance, you can measure the receptor strain just by with a range of biobit concentration and waiting until it reaches equilibrium, right? And then you take measurements, and then there you have your values, and you can get your threshold 5 and 95. Okay, what if, as I tried to do, but for me, because it wasn't a good approach, trying to infer or indirectly measure the emission constant mixing a 100% characterized receptor strain. In that case, I need to temporal and dynamically characterize the receptor strain. And that is because that is the reason after this plot that is a surface, I did several measurements on the receptor strain with the temporal dimension, and use this approach for being able to characterize the emission constant. But it's it was the things that it was the thing that I was available to do, what the available approach to do, but it was far from perfect. But it required to develop these formulas.
Okay, good. So that leads into the following question, which is already in chapter 4 for the experimental things. You estimated some parameters. You did a nice work on estimating parameters and so on. With those values that you estimated, did you rerun the models, or did those values for the parameters inform the original model in the second chapter? Or?
No, the problem is that sadly, the easier one to, okay, you had two problems here. First problem is that I considered, okay, if I wanted to measure my parameters, and I wanted to use the receptor strain to measure the parameters of the emitter strain and the sink strain, I then needed to start characterizing the receptor strain, okay, and then move forward with a really baroque approach for measuring and deducing the emitter. Now, the emitter strain and the sink strain are the ones that have a big impact in the shape of the white space, and I wasn't able to measure, for instance, properly measure the emitter strains, because it is shown in the thesis one example, but I warn the reader that this is an approach that it's worth not to try, and recommend the reader to directly measure the emission using another approaches like mass spectrometry for tiny molecules. So I wasn't able to characterize emitter and sink with indirect methods. And I don't recommend that to anyone.
But nevertheless, you estimated some parameters. Right?
Yes.
At least for the standard. So did you go back to the equations in chapter 2 and put those parameters instead of the ones you already did?
I did it to see what would be the shape. It's not showed here because I didn't have the measurements of all the strains, the three strains. But the problem is that, well, sink strain has two parameters, and I wasn't able to measure sink strain.
Okay. So yeah, I have one last question.
Okay.
When you talk about a fluorescent per cell or per unit cell, right? That would be the value that you would like to see for all the receptors, right? The fluorescent per cell. So did you thought at some point to go to use a flow cytometer to get the value per cell, not just try to estimate the population fluorescence?
Yeah. When I was doing the experiments, I took a few visits to the flow cytometry, and I did some tests, but the guys in the flow cytometry facility, they told me that it's a problem to measure bacterial cells, single cells, because they are so tiny that go in clumps. And also it would require a different approach that I'm going to share now. If you want to measure, for instance, green fluorescence is going to be your output, okay? And you have this receptor strain that is going to express in the presence of biobit green fluorescence, okay? And now you can somehow gate the green fluorescent receptor strain, okay? But we have a problem. If you do not observe clear fluorescence, does it mean that they are not induced or not? So you should put two different. One of the approaches was I should put two different fluorescent proteins. One of those, for instance, M-cherry. Okay, M-cherry is going to be expressed in the receptor cell, but in a constitutive manner. So the red fluorescence won't change won't depend on the bit concentration. Now, if I would use these red fluorescence for gating these cells, the receptor cells, and then measure there the amount of green fluorescence. And I just was finally tuning the experiments with the genes that I've shown in the presentation. So yes, I've thought about the possibility of measuring with flow cytometry, but I was focused in the presentation. Yeah, but all the approaches are here.
Okay, thank you very much. Well, I don't have any further question. Thank you very much.
Thank you. Thank you very much.
All right. Thanks, Angel. So we proceed now with the next member of the committee. I'm probably going to mispronounce your name. Sorry about that. Hyun Yok from University of Massachusetts Med School. It's your turn.
Yeah, thanks, Ricard. So Carlos, thanks for inviting me to be on your committee. It's it was very enjoyable, and the thesis itself, I like the fact that you were very honest about why you did it, why you did what you did, and what didn't work. And I really liked the honesty, and it really shows that you did this because this was really your idea. I'm sure you already did things too, of course, but you know, I can really see that this was something that you really cared about and passionate about, and you did it. And I have to say the thesis, certainly when I first got it, and when I still look at it now, it looks like because of the purple background in this triangle, it reminds me of this cult in the 1990s, religious cult. They didn't end up very well, but I'm sure you'll end up quite well. Yes, they wore this purple clothes, and they're no longer here. Let's put it that way.
I see. I didn't know about that. That's fine.
But I don't think they had a triangle, but purple was their thing. But anyway, okay. So indeed, you end your. So I want to start with something a little bit more, maybe general or not philosophical, but kind of more of a broad view, especially since you end your thesis with, this is a place to start, and you think about very general things. So it's in the title of your thesis, right? Computation. So I want to kind of discuss that. I want to hear your thoughts on just computation in general. So can a real cell, or your biobit cells, even if all the experiments worked out and everything, can they ever really be real Turing? Are they Turing complete?
Well, I actually remember there's a research group in Sevilla, my birthplace, natural computing by Mario Pérez Jiménez and George Paum, a Romanian guy, and they both developed this approach of membrane computing, in which they use this this framework in which you have substances encapsulated in different compartments. You can fuse the compartments. You can move substances from inside a compartment to another compartment, and you can add some operations inside and outside all the compartments. And they were able to prove. I'm not going to discuss the Turing completeness, because I'm not really an expert of what that means. I have notion, but I know what the difference between P and NP problem. And,
okay, yeah.
And they were able, this Mario Pérez Jiménez and George Paum, they were able to prove that, mathematically prove that some problems are NP, okay, that require non-polynomial time to be solved. They could be solved in polynomial time if you can make a computer based on this membrane computation. Of course, it would require it would decrease the computation time, but if I remember well, it would increase the number of membranes. So you are decreasing time complexity or time emptiness for you would be substituting that for space or number of membranes, and that is the limitation. So you change the nature of the limitations. It's still it's still a problem that needed to be solved. These problems, I don't know right now, didn't to be solved in non-polynomial time, but instead of time, they're going to use the size of the computer. And they were doing some simulation using these big NVIDIA graphic cards, and they had some good results. Results that show that at least this membrane computing paradigm can show qualitatively different behaviors from a regular computer. And they discussed how this is because the massively parallel approach, instead of, for instance, I don't know, my computer is, how many cores? Now that I'm in front of my computers, I think it's, I don't know, let's say it's 8 cores, right? No, it can be, whatever. It is 8 cores, let's imagine. Okay. So it can solve simultaneously 4 operations at the same time. Massively parallel means that hundreds, millions of operations at the same time. This is something that is going on right now in our brains, for instance. This is going on. And membrane computing, one of its features is this massively parallel approach. So we know that we are inhabiting machines, biological machines that they have this property of massively parallel programming. And I don't know if I'm going too philosophical here, so you can stop me at some point if you want.
No, but it's okay. Yeah.
But the idea is, yes, I think that some other alternative approaches using biological computers, whatever it means, because there are several approaches to biological computation, they may present some features, that make them compute in a qualitative, interesting and different manner. And that means that maybe some problems that are hard to solve, even with the best supercomputer, can be solved using a biological computer. But now, of course, we have the quantum computers. So why do we need the biological computers at all? That's just the final question, because we are discussing part of the thesis.
Okay. Well, let me just continue on that topic. So you mentioned basically time to finish solving a problem, right? That's what you're kind of talking about. And I want to go back to your implementation. So BioBit, and in general, like this quorum sensing type systems that Ricard and others have used. And I want to sort of think about, like, more formally kind of with you the complexity of a problem, right? So as you know, in computer science, like, in a physical computer, you measure as the input size gets larger, how long does it take to finish solving the problem, right? And that determines whether it's P or whatever, right? Or how would you determine the input size in your case, right? Because you have 1 bit, right, at the end of the day.
Yeah. Well,
how would you determine computational complexity in, like, a biocomputer?
Well, actually, I think that for being able to answer these questions, the question that you have proposed me, I don't have enough information to answer that honestly in my thesis. Okay.
Yeah.
What I was looking at is that everybody was so excited about developing logic gates, biological versions of logic gates. I'm excited with that, right? And people moved forward to implement distributed approaches to some specific problems. I list some of them in the thesis, and they are just they are just replicating digital electronics in most of the cases. So if we go through that path, then you can expect the complexity to be measured in the same way that we measure the complexity of programs or problems that run in these architectures. Okay, that is what I think. This is what I think. I think we lack. I think we lack, and I mentioned it in the last part of the book, in the last, funny chapter of the final question. For instance, I say biobit, and I put the B in front of the bit, like the Q in front of the bit from quantum bit, right? Because what is the thing here? The thing is that, for instance, qubits, quantum bits, they can acquire a value of 0, a value of 1, and a value of 0 and 1 simultaneously, right? Whereas the bits are running right now here in my computer, in your computer, they can be just only 1 and 0. Okay, and all the quantum computers and all that stuff is just a way of implementing operations that could be thought that exotic or not over this abstraction of what a quantum bit is. So I think that the idea of having a different bit, number one, with its own rules, and number two, something that can perform exotic operations, or that we consider exotic just arbitrarily, to this new bit, okay, that's important. That is my bet. I think that that's important, is an important concept. The other stuff is implementation. And for instance, a biobit can assume values from 0 to 1 in a continuous manner. That is not surprising. I've read that this is not something that is, oh my God, that's another whole different story. No, it's not so different. But I think that it could be interesting, the fact that, for instance, if you use wires that are placed in the buffer region, you can store temporal information there, right? Just imagine that you go inside a room and you switch off the light, and the light doesn't switch off, but you start counting the seconds. And as soon as the light switches off, right? 20 seconds. That means that this switch was turned on, like, I don't know, 2 hours ago. So that is something that you can implement with biobit, with this specific wire architecture. And I'm struggling.
But can you not implement that with a computer? You get a timer with a regular computer too, right?
Yes, I think you can. Right. Yes. And actually, it's something I would like to do, to see what happens.
So maybe, but you maybe just to continue the discussion, you mentioned something there. I think you hit on something, which is like the classical computer in front of you, or the quantum computers, whatever they may be. They are forced to use bits, right? It's a spin or whatever, right? So yes, it is binary. But whereas, as you say, your biobit, I mean, you call it a bit, but there's a concentration, right? I mean, everything that you're measuring, including calculating, is analog, right? So do you think so what kind of things can you now do if you don't think of it as just a bit, if you don't just force like a threshold and say, it doesn't respond or it responds, but rather it's a continuum, right? So what kind of things do you think it opens up, like in terms of the kinds of problems that your biobit computer can solve that regular computers would have a more difficult time solving? Or maybe just to put it another way. So I mean, it seems like, do you think that it's maybe just to say, like, do you think it's the wrong approach to put a threshold and say it's either on or off, when in fact your biobit is a range of it can attain a range of concentrations, and it's analog?
Yes, actually I think that the thresholds 5 and 95. This is arbitrary, and I always say that that's arbitrary. I think it helps us to set to conceptualize what is going on there, but it's arbitrary. You can actually take advantage of this continuous response. Yes, but fundamentally, again, you are asking me the final question, like, what is the purpose of this? And that leads us to what are the types of problem that could exclusively and efficiently solved using biological computers. And I think I have to I have to admit that I don't know. And I don't know if most of the people that are working with biological computers, from the not from the computation, from the, for instance, the guys of the membrane computing, they did a good job. They found a problem that could be solved with membrane computing, right? Another story is the implementation. That's another story. But I've been thinking about something, and I'm struggling. Every week I think about that. I've written to a couple of guys to see if they are thinking about a problem that could be solved, or even the different abstraction for bit abstraction that are there, right? To see if, hey, maybe one of these abstraction could be implemented. But I sadly have to say that I cannot come right now with a solution like this problem. This is one problem that if we implement the biobit with connect with this wire architecture, that will be exclusively solvable using this type of approach. Sorry, I'm afraid that I can. It's a question that I think is going to be attached to me.
Okay. Also, I don't want to take too much time, so I only have just a few more questions. And so maybe this is, now we're getting maybe a bit more specific into some details. So, so I like that for the theoretical part, I like that you state your assumptions. There are only two, and you define the equations, and then you just roll out the consequences. And that's right. That's the theoretical work, right? There's not any like additional things you throw in there in the middle of the work. So I really like that you're following logical consequences, and then you have mathematical formalism and so on. That's all great.
Thank you.
So, and you talked about standardization and, well, more for experiments, but in that spirit, so what you presented is a very beautiful theoretical work, and that's but on one hand, it is an implementation, right? You are thinking of some generic molecule, but it's a particular implementation. But in the spirit of this sort of the mathematical thinking that you have outlined, what is actually generalizable? Let's say if you have two different molecules, so two different biobits, what are the sort of the results that we can now generalize to n biobits or 2 biobits? So for example, what would it mean if you have 2 biobits? Does that mean I have 2 separate triangles that don't talk to each other, or is it one regular n-gon, or yeah? What?
Yes. I thought about that, and there is a word that is not here because I wanted to close a story and not do like, hey, a new different story appears in the middle of the thesis, which is even more abstract. If 3 strengths are required to build a wire, and the relative frequencies has to be constant, and that defines a triangle. And yes, Angel is it's right about the necessity of the representation of a triangle. Yes, you can put some, you can show some variables that take advantage of the triangular representation, but it's not the case for convergent divergence. What if we connect 2 wires, meaning that the receptor of one wire will be simultaneously the emitter of another different wire, another biobit, right? So the receptor of biobit 1, instead of expressing GFP, is going to emit a second biobit, right? In that case, we have 5 strains. That if we are operating in stationary phase conditions, the sum of the frequencies of these 5 strains remaining constant defines a triangle in higher dimensions, a simplex. Okay. So now you can actually you can extend, you can generalize what happens here just solely connecting wires without any logic in between yet. You can generalize that, and there's something that you can say about connecting wires using this framework. The ratio of each of these strains, if you start connecting one wire, another wire in a linear manner, doesn't matter, okay? The ratio, the proportion of each strain is going to decrease necessarily, okay? And you always are going to have one, if you have n strain, capital N strain, so you're guaranteed to have at least one strain with a ratio smaller than 1/n.
Okay.
Okay. And that is a problem because that is a problem for the wire in which that strain is participating as an emitter. Okay, as an emitter. Why is that? Because if you take a look at the time dynamics, okay, of the wire, I don't know where it is right now. Where's this? Sorry. Well, the idea is that this is going to impose a bottleneck on the time that it takes for a wire to turn on, the turning on time.
Okay.
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 Meaning that if you start connecting and connecting wires, and this is all liquid of, I don't know, 100 wires. Imagine that we move forward with the quantum sensing systems, and someone develops the final technique for developing synthetic wires. Now we can have any wire, and they do not do this crosstalk thing. So everything is orthogonal. It is ideal. Well, there you have another problem, which is if you want to put all these strains, 100 wires, for instance, connected.
Okay.
You know that at least one of the strains is going to be in a ratio, in a proportion with respect to the whole population, that is going to affect and is going to impose a bottleneck for the turning on and turning off time, meaning that the information flow is not going to respond to small inputs, to inputs of arbitrary duration. It will require this complex set of wire connected. They would be responsive for a different time scale. Okay, so it will require like bigger input time for the information to flow. That would be the idea.
Okay. All right. I just have 3 questions, two very quick ones, both about experiments. So you're doing these measurements on TCAN at a stationary phase, right?
Yes.
And you see that in some cases in the Davis media, the OD is constant, so it's a stationary phase, and the GFP is being produced. So first question is, how do you know that even though the OD is constant, that just means that the cells are physically intact, so it's detecting particles, the machine, but some of them might be physically intact and dead, right? Does that matter for you?
Well, this has been reported by DEFEND from Natalie's Balaban group in 2014, a work that I mentioned in the thesis, that that less than 5% because they were inducing cells in a stationary phase.
Mm-hmm.
They were inducing cells in stationary phase, and was in their paper they report that less than 5% of the cells present in your culture are growing. Okay, so you have super. In that case, what you expect. Sorry, with a doubling time of 20 hours. So actually, the renewal rate, it's almost negligible if you expect your wire to work in, I don't know, let's imagine 1 week, so to say.
Okay.
With the particular issue of the death of the cell death, well, if it would be something noticeable, a important phenomena, meaning that, for instance, 20% of the cell will die, maybe I may be wrong, but I would say that I should see that GFP decreases, and that's not what I see. Right? Once it has reached equilibrium. Because,
well, I mean, GFP can stay the same, right? I mean, if it doesn't degrade very fast and cells are not dividing, it could be a dead cell that still has GFP, I guess, right?
I agree with that, but it turns out that I put LVA degradation tag to all my GFPs, which does not work the same in exponential phase and in stationary phase. I thought it would be not really a big issue, but it turns out that the enzymes responsible for degradation, right? They increase its concentration in a stationary phase like 10 times. And proteolytic degradation that depend on these LVA tags gets more aggressive in a stationary phase. So that is an issue. That is not an issue. That is a feature actually that in this case, for answering your question, like these GFPs, they have they have short half-life because they are constantly degraded at a much higher rate than if they were in stationary phase because of this. So yeah, I would expect a decrease in GFP fluorescence. If that would be the case.
I will, for interest of time, I'll just ask one final question, and it's and I'm done. I'm sorry for taking time. It's very interesting work. So you know, I just want to hear your thoughts. So back, you know, so the issue of stochasticity was mentioned by Angel earlier, and I want to touch on that, which is, so you have to work with, as you said, a fairly large, macroscopic volume of millions of cells, right? But in the end, it's 1 bit. I'm not denigrating the work. It's very interesting, right? But it's 1 bit, lots of cells, you know, millions and millions of cells, large volume, very complicated in a way to implement. And yet you would expect that in a physical computer or whatever quantum computer, if you increase the number of players that are computing, you would expect things to, you know, you would increase computing power. And yet that doesn't seem to be happening here, beyond the fact that if you have more, you know, if you don't have 10 cells, but rather if you have millions of cells, okay, you can just take the averages. Are we doing something wrong? I mean, maybe we should be looking at, do you think we should be looking at 10, like population of 10 cells, or in some intermediate regime, and somehow the stochasticity is something that, you know, is something we can, you know, utilize for our own advantage to increase computing power?
Okay, I think that when you mean increasing the players in in quantum computers, for instance, or in regular computers, do you mean increasing the, for instance, the logic, the transistors and logic gates within the processors?
So that's a good point. So computing power is ill-defined here, right? So because in a way we haven't really defined like computational complexity, and we have to talk about input size, which is not the same as concentration, I think, of a bit.
Yeah.
Or maybe it is, right? If you frame it differently. So I just want to sort of hear your thoughts. That basically what I want to hear your thoughts on is it's so darn difficult to implement this thing, right? So the question is not why would you want to do this with cells, but like what, yeah, just sort of like, do you ever envision a case where you don't have to have so many different cells to implement a logic gate, for instance?
Okay.
Right. Experimentally, you're taking averages of millions and millions of cells, right?
Okay.
Do you ever see a scenario where, in fact, you can just have, let's say, 3 cells, 1 emitter, 1 receptor, 1 sink, where that would lead to a meaningful computation of implementation of a logic gate or buffer or something?
Well, certainly there are some scenarios in which you can make new features appear if you add noise to your system. Another story is if you want to take advantage of such features, but the classical one in two-dimensional phase plots where you have like a damped oscillation that converges to a fixed point in the space. If you if you add some noise, you are moving the system away from that point. So now that your point is in a vector field with forces to rotate, but if you get closer to the fixed point, then you're going to be moved again. And at the end, if you add some noise to a damped oscillation, you are going to start seeing oscillations. So meaning that if you, for instance, you implement some kind of systems with using this distributed approach, and this system, you have programmed it to show damped oscillations, right? With some specific strain combinations. Now, if you start decreasing the amount of cells, so, instead of millions of billions of each of the strains, then you start having maybe hundreds or 10 cells per strain, maybe even just one, but a few number of cells per each strain. Then at some point, what you expected to be a damped oscillation would become just an oscillation, right, around the fixed point. If you can get some profit using that new feature that appears because of this circumstance, that's another story. And I don't really have more intuition in the advantage of noise. I am aware that that is a thing. Okay?
Right. And maybe there's something, I think you are kind of on the right path. Maybe there's something to be said for if you have few cells, if you time average, you need more time, but you can have few cells, and that would maybe be the same as population averaging for a shorter amount of time. Maybe I think maybe you are on that path, but
yeah.
Okay. I think I took up so much of your time. Thanks a lot.
It's okay. Thank you, Yum. And
okay, sorry about that.
Okay.
Right. Thank you. Thanks a lot. Well, from my side, I have just maybe a few questions because you guys have done several of them, and then some comments. Okay, one of the things that I agree with Angel, it's about noise. That in a way, it's kind of. I understand that you cannot do everything, but it's a pity that you didn't explore noise as explicitly, because actually the nice thing of your work, in my opinion, one of the nice things is that usually we talk a lot about digital versus analog, but we don't go much farther from that. Usually we avoid to actually going into the real problem. And in your implementation, your formalism, you actually do consider that, where are the digital domains, etc., etc. So it's a pity, in fact, that you don't take noise, because noise kind of makes the analog computation brittle. It's the potential of the analog is is great, but you can go too far depending on what kind of computations you want to do. And it's very likely that the success of, let's say, cognitive agents in the biosphere is because the invention of the digital stuff, that you can actually propagate signals far away in a more reliable way.
Yeah.
So you didn't consider that. And also I wanted to ask, since you are a mathematically inclined person, why not to consider maybe, I know it's a generalization, but the general form of the degradation part of the biobits. It's a Michaelis-Menten, but you could have assume a general functional form. Say it saturates, it's 0 when B is 0, and try to give a look to that. Did you consider that?
Could you please develop a bit more? I want to fully understand.
You have this equation for the dynamics of your biobits concentration.
Yes.
You have a source term and a sink term. And in the sink term, you have a function that is Michaelis-Menten.
Yes.
But I don't think it would be, it's easy to say, but I don't think it would be difficult to actually try to make a bit more general this, and say, instead of Michaelis-Menten, I assume a function that saturates, right?
Okay.
Form, and it's 0 when B is 0, so that yours is a specific case. Did you consider that, or you consider?
No, and that is a great approach, because I love, going abstract, and yeah, that is great. Yeah, there's like the minimal requirements for a sink is saturation, for instance, and being 0 when it's 0, and where you could jump from 0 to a fixed sum, but that is not interesting at all. That is just a linear term in the equation. No, I haven't considered that, and I definitely need to try that.
I think it's worth trying, since you already get as far as you did. So I think it's really interesting.
I would like to say now that this thesis it won't end here, because I'm going to keep developing this by myself now, and every idea that you've told me, I'm going to consider to try, and that one of exploring different abstraction of different formulas, functions for the degradation term, that is something worth trying. Yeah, thank you.
Okay. I have a few general questions, comments, sorry, that I think are worth. In the introduction, I like that you are not just limiting yourself into the literature of the last 10 years, as most people do, just appear in Nature. You mentioned Leduc, for example.
Yes.
A really big precursor of what we can call synthetic biology. He uses that term. I will just as a point, I mean, because I've worked on that, and I think that usually it's too often ignored in synthetic biology. It's all the protocell story that some people in synthetic biology kind of ignores that this exists. But it's a particularly interesting domain here, because one interesting area within building synthetic protocells, which can be from really very much supramolecular chemistry, or to reduction, use vesicles with ribosomes or whatever it is, it's nice because there's a whole area now developed mostly by Keita Tamala and a few other people, where you actually have self-sustained cells that cannot replicate. You can engineer a lot there. You can engineer almost everything, and they can communicate, for example. And it looks like a very interesting place where actually the formalism that you're proposing could be applied, because it's pretty much under control. There's no growth of cells. They live a long time, and you can actually do a lot of stuff. And I think that it's interesting.
What is the name of the author? Sorry, again.
It's Kate Adamala. I can send this information.
Okay. Well, that is amazing. I've actually thought about the possibility of forgetting about cells, living organism, and go with cell-free systems in which you define like an arbitrary concentration of the your enzymes to be the maximum concentration, and you just play with the ratios there. And I thought about that, but this possibility of protocells that they do not replicate, that makes the thing more interesting. Yes. Yes.
Yeah. No, and the cell-free scenario also I think is interesting, and also in many ways, I don't know what Angel thinks about that, and unfortunately we cannot discuss that in person with a coffee because the situation is.
Yeah.
But is a big issue here that we have to consider when we talk about. I had my own question about Turing completeness, but you already did that. But the question is, how far can we go from in the analogies for complex computations? Because computation, when you implement that in machines, you usually have some clock that allows things to be synchronized, and that's a non-trivial thing, and that's something that may actually impose serious limitations on what actually can be computed by biological systems. I wanted to ask a very general question, but you don't have to make a very long answer. So do you think biology is programmable? Because that, around all the questions in synthetic biology is how far can we go actually to make things do what we want? And the programmability issue, what do you think about it? Just to know your opinion.
Well, actually, when we, in the introduction, it is shown a logic gate. So you can program a cell to act as a logic gate. Mm-hmm. But you know that, so your definition of programmable is another thing. You mean up to what point, at which point can we program cells? Is that what you're asking?
Well, you can see that in two ways. One is you can think in terms of the hardware and the software. But I'll give you my point, because maybe we can actually see.
Yes.
There's a contrast here. There's a lot of discussion always to say I'm going to program cells, and meaning that I'm going to tweak things there, cables and like a machine, and somehow I will make the cell do what I want. But that implies, in a way, thinking like an engineer, and that you can actually act on biological systems, whether or not you can do machines, as something that you can modify as much as you want, right?
Yeah.
Whereas, is that the case really, or we have a chassis we don't understand, and we put a minimal amount of things there so that the chassis doesn't break up and
that is actually that. Okay, now I understand, and well, that is actually a problem when if I write a code that is running on my computer, it turns out that my computer has been built by engineers and not me, not even an arbitrary informatician, but there is a guy out there that knows how this thing works. Now we have a problem with synthetic biology because we build our fancy circuits to do whatever we want, and the computer, the machine that is going to run this program, it happens to be an organism that is actively, it's a matter of research, right? Even E. coli. E. coli is the most studied bacteria easily, and we don't fully understand some of the features. There is there is research in the like, so that is a problem. That is actually a huge problem. So in that case, I would refine the question to say, can we reach a point at which we can be sure that any arbitrary circuit that we could implement in a cell won't throw us some new interaction with the host? So equivalent to, can we fully understand the host organism? I don't know if we will be able to do that, but there's the research in synthetic cells. So synthetic cells, minimal cells, that is more in the line of protocells that you have mentioned. Maybe we can have a chance of program whatever thing inside a cell. But there is another issue that it's pointed out by this paper about the cellular supremacy, by the way, from the Goñi Moreno's group. And they say, well, living organisms, they have some unique features that we may want to use as an advantage. One of the things is evolvability. Well, but that is another thing that you need to control at the same time. And I see more something that I don't really know if. So one problem is that, for instance, if you want one limitation would be, if you want your circuit to behave in a robust manner across generations, then you're going to have a problem, right? Because evolution may negatively affect your circuit. So yes, but we need to take advantage of that. But I think evolution in that sense could be a new problem, analogous somehow of to the problem of fully understanding an organism. So if we put a program there, we can be sure that there is not going to be an emerging behavior that ruins all our programs. But okay, but now I have to do the same with evolution. And I don't really know if I think we can find a good space, a big space, a safe space within synthetic biology in which we can place our organisms and we can play with them, but we will always be a little bit worried about if this is going to not work because of some new things. I think that this is going to be inherent to programming living systems, I'm afraid.
Okay. Well, we definitely need a coffee table to keep discussing this kind of thing.
Yeah, for sure.
Okay, sure.
So just a final comment. I also think that this kind of a serious exploration of the wire, as the way you do, it connects with other problems that may be worth exploring. One thing that came to my mind is there's been some theoretical efforts also in modeling what some people call, I think, is astrocyte modulated synapses. So what happened in the brain when you have neurons connecting to each other, it looks like the glia cells play some more role than just being supporting, etc. So they help actually in the computations. And there's some modeling efforts that have been published a few years ago that might have interesting connections with your work. Because what essentially people says is, see, I've had two neurons and the communication between two of them is modulated by the third partner there. So I think it's an interesting theoretical connection worth exploring. And I have to say that reading your dissertation has also been inspiring me, because I'm these days thinking in this liquid brain story. So, you know, how will happen if you have a computation in a system where the agents do not maintain topological connections over time. And it's really interesting, because one thing that seems to be very universal is that to do some computations, you need interneurons. So things that in a way have to do with considering wires that are not just connecting a signal from a receptor and making it do an action. As soon as you have that, you can actually make a big jump. And that's not available to everyone. And your formalism kind of helped me to think about that. So just wanted to
Thank you.
right that
I'm
and
glad that it worked for you.
yeah, yeah. I mean, thanks. I remember years ago you came to my office and you explained to me your idea of the biobits. You were very excited, and I'm glad that you actually completed this part of the work. And also, I agree with what Kim was saying, that you've been honest in saying this is what I have done, and there's this open new directions. I know from Jordi that you have other things to complete. The e-science paper is really, really nice. And yeah, my congratulations for what you have done so far, and I hope you keep pushing this in new directions.
Definitely, definitely. Thank you very much.
So usually in these meetings, when there's people around, real people, we ask if there's someone in the audience, some PhD person that wants to say something, or from the supervisor.
May I say something, please?
Sure.
Okay.
Go ahead.
I would be more than pleased if some of you has a question to answer that. Okay, that's what I wanted to say. Thank you.
All right. Okay, if there are no more questions, thanks again. Jordi, you want to say anything?
No, not much, especially just, I mean, we could continue talking about the questions that you have been making, which I find fascinating, all of them, but I think we should not take too much time. I would just like to thank Carlos for being in the lab for a few years and lighting it up, making it brighter, and I'm sure that everybody was very happy.
Thank you so much to you, Jordi.
And you have seen what he has done, so there's nothing else to say, so thank you.
Okay. All right. So in that case, the people from the committee will get together into the private story. Do you mind, Jordi, to join us just at least for a few minutes, if in case we need to ask you anything?
Yes, sure, of course. I will join you right now. I have the link, so I'll go to. So let's go all four of us. Then we go to the other link. Okay, yes. Great.
Yeah.
Okay, thank you.
Okay.
Thank you.
Good. So the rest of you stay here, eh?
Yes, we have to stay here now. Okay.
Yes, you stay here, and then I go, and then I'll come back, and you do whatever you want. Just laugh, talk. See you in a few minutes.
Hey.
Hello to everybody that is watching this in Zoom, in YouTube, and also in Twitch.
Well done.
Thank you. Oh my God. I've enjoyed it. I've enjoyed it a lot. He disfrutado mucho. Wow. Yes. I didn't know the purple thing with the cold. That is weird. I'll look for it. But yeah, they've told me that this looks like a conspiracy weird thing or whatever. But I like it. I like the triangle there. It's been years with this thing. I've enjoyed it. I hope you have enjoyed it. Don't leave right now. Well, you're not going to leave, but there are more people watching this defense in YouTube and en Twitch. No os vayáis si estáis viendo esto en YouTube y en Twitch, porque ahora viene el momento de la verdad. Ahora viene el momento de la verdad, ¿vale? En, 5 o 10 minutos. And oh my God, have you enjoyed? You can say something.
It was very nice, Carlos.
Thank you. Sorry, you can stop laughing now.
You can release all the stress now.
How can I change? Okay, the view. Galeria. Okay, now I'm seeing all of your faces. Thank you so much. for the people that is watching this from another continent with a different hour, Julia, Rosa, and especially Leticia, which is in the west coast, and it's even
For
worse.
Leticia, I think
Look at that. It's nice to see you all of here. Oh my god. Oh my god. Well, they seem to have enjoyed. I was afraid that I possibly you don't know, but the way the thesis has been written has a friendly language, and they seem to have enjoyed. How about that? I was afraid that maybe that wouldn't be the case. I will make it available online at some point, so you can just have it there. Okay. By the way, once they come again and say whatever they have to say, you can, put your mics at the same time. Okay. Okay. Say something. This is weird. It's just me talking alone.
You are used to that.
Yeah, that is true, but it's still weird. Now there's a lot of people watching my face.
Carlos, we've never been in a stream before.
Sorry?
And is Luis your brother?
Yes, that's my brother in the grid. He's responsible for broadcasting this in YouTube and Twitch.
That's why everybody's so quiet. No one has been in a broadcast before, Carlos.
Well, that's kind of a broadcast. This is open. Yes, possibly. But, well, now everybody can see the thesis, right? Oh my God. Well.
What are you going to do afterwards?
I'm going to spend some time, relaxed. I don't see myself going for a postdoc, actually. Something that I know.
I think today.
Today.
What are you going to do right after?
Well, I think I'm going to go. My parents came by surprise. I didn't know they were gonna come. There they are, and I think I'm gonna go out for lunch with my partner and my parents, and yes, and what the fuck? Sorry, what? Lorena Espinar is at home right now. I don't understand anything.
Ves afuera de tu habitación y saluda a tu familia y dales un abrazo. Hombre, Carlos, tanta historia.
But they're going to come right now, right? Okay. Give me one. Give me just...
Tanto streaming.
Wait a second.
Ves con tu familia.
Marcel answers anything for you, don't worry.
Ostras, Marta, yo.
Hola, Marcus.
Que estava al YouTube per donar-li visites al Carlos i ara m'he passat aquí per poder-lo felicitar.
Okay. I'm here.
Hola, qué silencio, qué silencio, no sé en qué.
Sí,
idioma.
verdad, es muy raro.
Ay, mira, no conocía a tu hermano. Hola, ¿es tu hermano?
Sí, es mi hermano.
Ah,
mi hermano.
Mucho gusto. Ahora nos vemos. Bueno, ya he hablado con el tribunal. Uy, cuánta gente hay aquí. Vale. Bueno, me callo.
Ok.
Esto es un poco, hay mucha gente. Bueno, y a tus padres también, hola.
Sí.
Me alegro de verles. Muy bien. ¿Sabes lo que falta aquí? No hay manera. Yo les intenté convencer de que hicieran breakout rooms, porque es una pena. A lo mejor haciendo breakout rooms la gente podría entrar y salir o algo así, pero bueno.
No, está
de
bien.
Manera.
Sí, sí.
Bueno, veo a Rosa, Marsal, Julia, madre mía. No sé si yo no veo más gente aquí. Leticia supongo que está por algún sitio.
Qué locura.
Mira, Lorena, mira.
Qué locura. Por Dios. Por Dios.
Bueno, ha ido bien, ¿eh? Yo creo que, ¿ves?
¿Lo has disfrutado, no, Carlos?
Se notaba que le gustaba, se notaba que le encantaba.
Lo he disfrutado muchísimo, lo he disfrutado muchísimo. Y no me he colado, porque compactar esto, acercarlo a los 45 minutos ha sido difícil. Bajarlo de la hora ha sido muy difícil. Y he tenido que sacar la guadaña y quitar ahí un poco todo. Joder. Pero bueno, al final creo que ha estado bien, ¿no? Creo que ha estado bien. Y ¡guau! ¡Qué locura! ¡Qué locura!
En tu caso tenía que haber hecho una tesis extra large. Un par de horitas de charla tuya y ya está. Y luego otras 2 o 3 horas de preguntas.
Te voy a decir una cosa. Para mí eso no hubiera sido un problema, ¿eh?
Ya lo sé. No, si lo digo. Ya lo sé.
No tener la limitación del tiempo, incluso ya ahora voy a sacar una pizarra y voy a coger y a escribir. ¡Buah! Claro. Eso hubiera sido totalmente ideal. Totalmente ideal.
Esto es lo que pasa en el Reino Unido. Yo creo que te hubieran gustado. ¿Hay alguien que haya hecho la tesis en el Reino Unido? O que la haya? Esto es lo que te hubiera gustado desde el Reino Unido. Ahí encerrado con el tribunal 2 o 3 horas o 4.
Lo hubiera aguantado, ¿eh? Porque
no, que desde luego.
Si me dicen, no tienes que controlar la duración, ¡buah! ¡Qué paz! Puedo meter absolutamente todo lo que quiero. Sí. Y las preguntas también las he disfrutado mucho, ¿eh? Las he disfrutado. Me han gustado mucho las preguntas y bueno, me ha gustado ver que hay uno que ha visto el paper del artículo porque se publicó la semana pasada. Muy bien. Y no lo dije, creo que, sí, lo dije creo que por Twitter. Pero bueno, yo qué sé, ¿sabes? Te digo que está bien. Está muy bien eso. Joder, qué locura.
Bueno, hay que meter ruido a esto, Carlos. Esto se nos pasó. No puede ser.
Hay
que meter ruido a las células. Aunque haya muchas células, puede haber...
vale. Por un momento Jordi estaba pensando en meter una música de fondo. Digo, ¿qué? ¿Un lofi bajito?
No ruido físico. Ruido...
Vale. No, sí,
perdón, me he ido a la ciencia, perdón. Lo retiro.
No, por Dios.
Vamos a dejarlo.
No, sí, la verdad es que es una cosa que puede estar interesante meterlo a ver qué es lo que sucede.
Porque si hay heterogeneidad en las strains, entonces la cosa puede ser incluso más interesante. Pero bueno, ya.
Hombre, por muchas células que tú tengas, si tú tienes un número suficientemente grande de diferentes strains, al final el ruido tiene que jugar algún papel ahí. No sé cómo lo he dicho esto en la presentación, esto creo que era interesante. Pero bueno, no lo he dicho, se me ha pasado. Y bueno, pues sí, tendré que ver cómo se hace eso.
Carlos, en los extremos de los vértices, donde tienes menos de un tipo de célula, ahí sí que tiene que importar, por ejemplo, el ruido.
Ahí sí. Llevas razón. Llevas razón. Muchas gracias, doctora Galera. Eso no lo has visto.
Y a esas horas que tiene que estar ya oscura porque si
Eso sí que es buena. Esa sí que es muy buena, de hecho. O sea, yo sé, por ejemplo, que no hay un gran crecimiento de células. La población al final, la mayoría está viva y la tasa de renovación poblacional es súper baja, como he dicho. Una división cada 20 horas y solamente sucede en menos del 5% de la población. Pero dices, bueno, ya, y si nos ponemos ahí a ver qué pasa, tú empiezas con un punto en el triángulo y tú dices, voy a empezar siempre en el mismo punto y voy a dejar trayectorias con esa tasa tan pequeña de renovación. Hay un área máxima a la que puedes llegar, que es como un hexágono. Si tú estás cerca de un extremo, sí que puedes tener un problema ahí. Eso es mejor mantenerse lejos de los extremos. Pero, joder, muy bien, bien visto. Muy bien.
Okay. No, Ricardo, está aquí el tribunal. Ha sido rápido. This has been fast.
Yeah. It's been fast, yeah. in part because the paperwork will be completed not right now, so it's not probably the only thing we needed to know. It's agreement. So it's the other people from the committee there.
Yes.
Yeah, I'm here.
I can see Angel and Jan, yes.
Yeah, I am here. Yeah.
Okay. Yeah. Okay, excellent. All right. So we agreed, all of us, unanimously, to give you the highest scores for your dissertation. Excellent, cum laude. Congratulations. Don't laugh. Don't laugh like you're like crazy. So,
you can open your mix now.
Congrats, Carlos.
Congrats, Carlos.
Congratulations.
Thank you.
Congratulations.
Thank you so much. Thank you so much. Thank you. Oh my God. Okay.
And we hope you keep doing that and you know, doing original work. Keep pushing these ideas, and wish you the best.
Okay, thank you so much, Ricard. Thank you to the members of the committee, and thank you to everyone that has attended here in Zoom or in the other platforms. To all of you, to my family, to my partner. Thank you all for being here in this day. Thank you so much. It's been a pleasure. It's been a total pleasure. And thanks to Jordi Garcia Jalvo, my thesis supervisor, for giving me the chance to be here and develop my ideas. Thank you.
Congratulations, Dr. Toscano.
Thank you.
A celebrarlo ahora.
Thank you.
Now let's go to the Pika Pika.
Yeah, I don't know the Pika Pika. Okay.
Mira.
Mira. Ay, sí, es verdad que se venga para acá.
Okay.
Oh, qué maravilla.
Well,
okay.
Again, it's sad that this happens in this virtual environment, but I hope you feel happy now, and we'll see you around.
Thanks, Ricard.
Thank you. Thank you so much,
Ricard. Yes.
Oh,
wow.
Vale. Okay.
Wow, beautiful timing.
Thank you, Angel. Thank you, Jan-Yuk, for being here, and thank you, Ricard. Thank all of you. And
okay.
I have a present.
Open your present.
I have to open here. Oh my God! Oh my God! So wait a second. I'm not hearing you right now. Finally, I have the triangle. I have my hand with the triangle and the three types of cells and the BioVid. I don't know if you can see it there. There is BioVid. So I'm going to wait right now. Okay. Sorry, I have a difficult yet.
You have to move the camera or something so that we can take a pictures.
¡Woo! ¡Bravo! ¡Qué bonito!
Thank you so much. Thank you. Muchas gracias. Es el momento de ponerme esto.
Se queda genial.
Muchísimas gracias por estar aquí. Muchísimas gracias. Gracias por haberme acompañado todos los que he tenido alguna vez en este trayecto. Y nada, ahora llega pues la siguiente etapa de ya por fin he conseguido el doctorado y ahora moving forward a lo siguiente. Gracias a todos, de corazón, muchas gracias.
Enhorabuena.
Felicidades. Bueno.
Is there something else happening? ¿Hay alguna cosa más? O espera, no sé si alguien quiere decir algo.
¿Cómo?
No sé si alguien quiere decir algo. ¿Hay alguna cosa más pasando? ¿Qué pase?
A celebrarlo.
They're there. I don't know. Leticia, Leticia's gone. Are you there, Leticia? I don't know if she wants to say something.
Leticia, se ha ido.
She's gone. Leticia, are you there? Is there something that we should do? ¿Hay alguna cosa que... ¿Cómo va esto? No te oímos.
We have a video for him, but I think we would prefer to have it when we are not in other platforms. I didn't ask for other people for permission.
Okay.
So maybe we can do it more privately. I don't know.
Okay. Yes. It's going to happen right now.
It's very personal, the video.
Don't worry. Luis, te voy a hacer una señal y cortas. Muchas gracias a los que estáis viendo esto desde YouTube y Twitch. Sí, esto ha sido retransmitido no solamente en YouTube, en Twitch. Probablemente sea la primera tesis que se retransmite en Twitch, lo cual es muy trash. Muchas gracias. Nos vemos próximamente. Un beso.